Greetings, future and current biotechies. My name is Soraya, and today we are going to talk about DNA microarrays. Now, first of all, we're going to go through this PowerPoint and we're going to ask 4 main questions. What is a microarray? How does it work? Where is it used? And how does it benefit us in science today? Now, first of all, what even is a microarray? A microarray is an instrument used in a scientific lab to detect and measure gene expression at a rate of thousands of genes at a time. They can identify the presence or absence of certain genes and can also examine how the genes are affected by external stimuli. The type of technology that microarrays utilize are made specially for analyzing complex biological systems. They are especially good at detecting genetic mutations in DNA. A DNA microarray is also known as a DNA chip or biochip. Microarrays are the collection of very tiny chips or DNA hotspots that are attached to a solid surface. The solid surface is usually a glass or silicone sheet that holds thousands of DNA chips or probes used to identify the genes being expressed. There are 4 main types of microarrays currently being used. They are listed as the cDNA, oligoDNA, BAC, and SNP microarray. The microarray is a scientific instrument that uses technology that was only recently refined and discovered around 20 years ago. Scientists and researchers both favor the microarray for its innate ability to read and analyze thousands of genes in a single take reaction and produce results that can be used in a controlled environment. Now we ask our second question. How does the microarray work? And is there more than one way for it to be used? Microarrays use a process called hybridization or binding. Hybridization is when complementary strands of DNA, or cDNA, are binded together. However, before hybridization can begin, the DNA needs to be denatured or separated from between complementary strands. The separated strands of DNA are then cut into smaller, more manageable pieces or oligonucleotides. These pieces are then labeled with a fluorescent dye. Depending on the process being ran and the mode of preparation, the microarray is classified into 3 separate categories. These categories are known as the spotted array on glass, where the microarray is made on glass-coated microscopic slides that are coated in polylysine. The polylysine is a special polymer that allows for adhesion of the DNA to the slides. The second category would be self-assembled array. This classification of array is fiber optic based, meaning the DNA is deposited onto polystyrene beads. The third category would be in situ synthesized array. This type of array is curated by a chemical synthetic process built on a solid substrate that is represented by surface tension. The chemical synthesis requires RNA primer, DNA polymerase, and an array of oligonucleotides. Keep in mind that oligonucleotides are short segments of DNA, usually single strands of DNA, that are less than 50 nucleotide pairs long. The oligonucleotides will adhere to glass slides in order to create the assembly used in the microarray. This classification of microarrays is specifically utilized in genotyping, sequencing, and gene expression analysis. Microarrays typically use an experimental sample, the information being collected, and a reference sample, or a control group that the results will be measured or tested against. An example would be the measure of healthy human cells measured against cancerous cells. There are roughly 6 steps to summarize the basic process of how the microarray functions in a controlled environment. Number 1, the mRNA strand is isolated from the DNA template. Number 2, the samples in the array are hybridized. Number 3, the microarray detects and quantifies the signals being released during the gene expression. Number 4, you identify and analyze the co-regulated genes and the related genes. And number 5, you perform a northern blot analysis to confirm the presence of regulated genes. And finally, number 6, you collect all of data and place into a databank. Remember that microarrays are excellent at identifying mutations in the DNA. If a mutation is present, then that fragment of DNA will be unable to properly bind to the sequences on the biochip that represents a normal sequence. The mutated fragment will instead bind to the sequence on the chip that represents mutated DNA. Our third question asks, where are microarrays used? Microarrays are not limited in the number of career paths that they are utilized. They are greatly used in the medical field, but also in forensics, agriculture, and toxicology. As mentioned above, microarrays are typically used in the medical field to quantify research based on human cells and cancer cells to help solve mysteries about human-related diseases and issues. Our last question, how does it benefit researchers and scientists today? Microarrays have become excellent tools to use in the workforce in aiding scientists and researchers in understanding more about our genes. They aid in the examination of diseases and can even provide insight into how to identify and treat diseases in our genealogy. Due to microarrays, scientists and medical staff now know more about many complex diseases such as diabetes, heart disease, cancer, and more. Microarrays have broadened fields of study for many industries, such as in medical research, drug development, and even food production. With the continued usage of this instrument, their microarray will open many doors for researchers to continue to identify problems in the DNA and be successful in developing methods to combat every obstacle. Now that we're finished with our PowerPoint, we're going to pan over to our current biotechie Tatiana, who is in a classroom setting, who will demonstrate how a microarray works using a SCME model of a lithographic microarray.
Hello, current and future biotechies. As Soraya has finished describing how microarrays in detail, to summarize what she said, microarrays is an extremely helpful tool to have in a medical setting or workplace environment. Microarrays use small DNA probes in the form of oligonucleotides to quantifiably measure how genes are expressed in hybridized DNA strands. There are several methods to how microarrays are used, as Soraya already stated. The way that we will demonstrate today is a photolithographic microarray, which will be represented by these 12 individual masks, and the masks are used as a template to form the oligonucleotides. Each mask has a specific requirement set up in order to link each nucleotide in the sequence. And as we can see, the black beads represent adenine, the green represents cytosine, the pink beads represent thymine, and the purple beads represent guanine. So before we start building our model, we have to know that there are 3 steps in between the masks. One is called a protect, another is called a deprotect with UV light, and the next one is called addition. So, knowing that, we know now that this is representing a silicone wafer that's already been protected with a blocking agent. So now we can start by adding the first mask, which is this one, carefully place it on top of this. And then we can use a flashlight to simulate it as UV light. And what the UV light is doing, it is deprotecting the base so we can, so the specific oligonucleotides, which are, in this case, thymine, represented by the letter T here in the color. And it deprotects it for 2 seconds, and then you can begin by adding on each individual opening. Just make sure everything, like, they go in there and that they're all in there. So now that we have that, we can additionally move on to the second mask. So we just move it. And in a real fabrication process, there would be a blocking agent that gets placed on top to protect the thymine. And so we add mask 2. And once again, we use the UV light to deprotect it. So therefore, the next sequence of nucleotide would be cytosine, which is green. And those get placed in the individual little openings. So there we have it. We've built our 8x8 silicone wafer representing a real microarray. And now each of the cells have 3 oligonucleotides that can be used as probes so they can link or hybridize with another single-stranded DNA. And now since it's formed and everything, we can discuss about positive and negative controls existing in all biological assays, such as the DNA microarrays. Because of the complexity of microarray fabrication, the purpose of positive and negative controls is to verify the overall performance of the microarray and the analytical technique. But since that's all the time we have, we can discuss that in another video. Goodbye, future and current biotechies.